Hello everyone, a very warm welcome to my botany classes. The topic for today's discussion is culture media for microbes. This is a very important topic for studying all types of microbes and their culture. Okay, microbes are generally allowed to propagate in culture media for the purpose of study. Okay, so how do we define culture media? Culture media are nutrient medium or preparations that supports and allows microorganisms to be propagated in the laboratory for further study. See, when a microorganism gets into the body of a host, suppose if you get common cold, that means what? The influenza virus has entered your body and has got a chance and has got a favorable ambience inside your body to propagate. But when those microorganisms are cultured in the laboratory, they don't normally get that atmosphere. In order to provide them with nutrients and items that support their growth, we need to provide them with culture media or culture medium. Culture medium or culture media are rich in nutrients. Okay, they are prepared in a special way that support the propagation of microorganisms for future studies. Okay, see, what do these culture media do? They provide all necessary nutrients and growth factors that encourage the development of the organism inoculated in it. Suppose you have a Petri plate like that. Okay, you have your culture media in this. The organism which you are trying to grow, the microorganism, is inoculated into this. Unless and until it is inoculated, how will it grow, isn't it? So it is inoculated here and it starts growing. It will only grow if it is having the favorable or the required favorable conditions. Okay, microorganisms are usually introduced into the culture media, which can be solid, liquid, or semi-solid. So culture media can be of what various types? Solid, liquid, or semi-solid, through a process known as inoculation. So this is inoculation, this thing. Introducing the microorganism into the culture media is called inoculation. Okay, and the inoculated plates or medium are incubated at optimum temperature and later observed for microbial growth known as culture. Okay, the microorganism, they grow to give you the culture which you use for further studies. They need proper nutrients, they need proper growth factors that will help them to propagate and grow, and a correct temperature. Not all temperatures are favorable for their growth. They need optimal temperature. Okay, so a culture in microbiology refers to colonies of microorganisms, particularly bacteria, that grow and multiplies in or on a culture media. So what is this culture? I just now told you the microorganism will grow in the media and will give you a culture. What is that culture? This culture is nothing but the colonies of microorganisms. Especially here we talk about bacteria that grow and multiply in or on a culture media. See, this is how it looks. Can you see these spots? These are nothing but colonies. Okay. These stripes here, these are all colonies. Now see, a culture in microbiology refers to colony which I just told you. There are several culture media available for the propagation of microorganisms as well as for the transportation and storage of microbes in microbiology. So, culture media are not only used for their propagation but also used for two other purposes. What are they? Transportation and storage of microbes. So, microbes are sometimes stored for future study. Okay. Now see, what is the general purpose? What is general purpose media? What is the basic idea behind general purpose media? It is nothing but simple or basal culture media. Okay. It is a routine culture media which is used for the cultivation of microorganism in microbiology laboratory. So it is the simplest type of media that you use in microbiology labs. They are basically used for the culturing of bacteria that do not need extra growth nutrients and are not fastidious in nature. Okay. General purpose media support the growth of a wide variety of bacteria and they do not contain any growth inhibitory substance. This is something very, very important. If it contains growth inhibitory substances, then how will the microorganism grow? Its growth becomes hindered. So when you are preparing a media, you should keep in mind that it should not contain any growth inhibiting factors in it. Okay. Another term you have come across here, fastidious. What is fastidious bacteria? Any idea? Student, what is fastidious? Do you know any example of a fastidious bacteria? A fastidious organism is any organism that has complex or particular nutritional requirement. I just mentioned here that, see, bacteria that do not need extra growth factor. What did I say? Bacteria that do not need extra growth factors or growth nutrients and are not fastidious in nature. That means they are not requiring extra or any complex or particular nutritional needs. They don't have. Or in other words, you can say a fastidious organism will only grow when specific nutrients are included in its medium. They are very particular. Okay, they're very, very particular. Like for fastidious bacteria, one or two examples if I give you. Hemophilic ones are Haemophilus species. These bacteria, they are extremely fastidious in nature. Prevotella species, these are anaerobes. That is also fastidious in nature. Okay. Commonly used general purpose media in microbiology laboratory include nutrient agar, nutrient broth, peptone water, tryptic soy broth, tryptic soy agar, Muller-Hilton broth, and Muller-Hilton agar, amongst others. So you will have to remember these names. What are the commonly used general purpose media? What are they? To name some of them, nutrient agar, nutrient broth, then peptone water, then tryptic soy broth, tryptic soy agar, Muller-Hilton. So you'll have to remember these names while you are learning the lesson and don't make spelling mistakes. These are very, very specific. Muller-Hilton or MH media, you can also call it MH media. Muller-Hilton agar is the best medium for conducting antimicrobial susceptibility testing, which is also known as AST, antimicrobial susceptibility testing in the laboratory. Okay. So MH agar is very much ideal or rather the best for AST, that is antimicrobial susceptibility testing. Fine. Now we'll talk about enriched media. These are usually liquid media or broth that supports the growth of a particular bacterium while inhibiting the growth of unwanted bacteria. Suppose you are trying to culture a particular type of bacteria. Okay, you take a sample of water or you just take a sample of soil. You cannot say that only one particular bacterium will be there. There can be many, isn't it? Many different colonies of bacteria will be there. Fine. Now, enriched media is very particular. Enriched media are very, very particular. What will they do? They will only support the growth of a particular bacterium and will not support the unwanted ones. Unwanted bacteria will not be allowed to grow on that media. Okay. They contain additional growth nutrients, example, blood, serum, and egg yolk, like enrichment media for the cultivation and isolation of fastidious bacteria. But unlike enrichment media, enriched media are mainly solid culture media. All right. Generally used to, these type of media are generally used to cultivate fastidious bacteria, example Streptococcus species, Haemophilus I was just talking about. Now, when you study each type of bacteria, two things you will have to know, rather three things. First is, what is it? That is the definition. Second is what type of organism grows. And third is what? Examples. You will have to know this. Okay. Example what? Selenite F broth culture medium. This is used for culture of fecal specimen. Okay, bacterial specimen in the stool. Other than that, other than selenite F broth, what do you have? Alkaline peptone water, blood agar, and chocolate agar are examples of enriched culture media. Okay. See, this is how blood agar and chocolate agar look. Next, we have selective media. Culture media that promote the growth of certain type of bacteria while inhibiting the growth of the undesired organism. Okay, this selective one, from the very word you can understand, they have got a selective role. They will select one particular bacteria and will reject the other. They will allow one particular type of bacterium to grow and they will not promote the growth of other bacteria. Okay. Selective media contain inhibitory substances such as dyes, salts, and antibiotics which prevent the growth of undesired microorganism. Okay. Example, mannitol salt agar. Here also same thing. See, you know the definition. Now you'll have to know the example. Mannitol salt agar which contain NaCl that inhibits some other bacteria, the non-desired bacteria. McConkey agar which contain bile salts and crystal violet that inhibit growth of gram-positive bacteria. So you can use this for what? You can use this for gram-negative ones because it will have inhibitors for gram-positive bacteria. Okay. Then comes Seborrhoeic agar, which contain antibiotics that inhibit bacterial growth, and tellurite media, which contains potassium tellurite that inhibit many bacteria, excluding Corynebacterium diphtheria. So you can use this media for culturing this bacterium, but not for anyone else. Okay. See, study this chart very, very nicely. This chart contains names of test organisms along with the selective media in which they grow. Okay. Staphylococcus aureus, they grow on milk agar. Escherichia coli, that is E. coli, they grow on what? They grow on EMB, that is eosin methylene blue agar. Pseudomonas, they grow on Pseudomonas agar. Shigella, they grow on deoxycolate agar, deoxycolate agar. Then Streptococci, they grow on azide blood agar. Staphylococcus aureus, they grow on MSA, that is mannitol salt agar. Bacillus subtilis, they grow on nutrient agar. Salmonella, they grow on Blair bismuth sulfite agar. Aerobacter aerogenes, they grow on EMB, eosine methylene blue agar, like this. Okay. Now coming to differential media. What is it? Growth media that allows certain bacteria to have distinct colonies on the culture media. Okay. Organisms growing on differential media produce characteristic colonies that differentiate them from other groups of microbes. Suppose a soil sample contains, say, 5 different types of bacteria. This is just an example to make you understand. Suppose this is a media plate. Okay. 5 different types of bacteria are present in the soil. You have inoculated the soil sample on the plate. Now see, this is one. I'm just changing the colors in order to make you understand. I'll use 5 different colors. This is one. This is one. And this is one. So by looking at it, you can understand this is different from this, this is different from this, this is different from this one, and this is different from this one. This is different from this. So by the very appearance of the colonies, you can understand that they are different. So they produce characteristic colonies. Maybe the morphology is different, the color is different. They produce characteristic colonies that differentiate them from other groups of microbes. Okay. They are also known as indicator media because they contain certain indicators. What are they known as? They're also known as indicator media. Okay. Neutral red indicates because they contain certain indicators like neutral red, chemicals, and dyes. All right. With changes in color, especially when the definite organism, that is the organism of interest, is present in the specimen being studied. All right. Now see, McConkey agar is a differential media that help microbiologists to differentiate lactose fermenting bacteria, that is E. coli, which ferments lactose to produce pink colonies on McConkey agar, that is MAC, from non-lactose fermenting bacteria, that is Salmonella, which does not ferment lactose and thus appear paler colorless colonies on the growth medium. So from the very color, you understand that whether this is lactose fermenting bacteria E. coli or non-fermenting, the bacteria which does not ferment lactose. Okay. Examples of differential media are cysteine lactose electrolyte deficient media or CLED media, CLED media, mannitol salt agar, McConkey agar, and blood agar are some of the examples of this particular type, that is differential media. All right. Now, finally, we come to transport media. What is that? These media are used for transporting specimen or microorganisms from one location to another. In the beginning of the lecture, if you have paid attention, I had told you that it is not only, the media are not only used for laboratory study purposes, but it is also used for transporting microorganisms. Okay, so they're also used in cases where the samples collected will not be cultured immediately after collection. Fine. That means what? It is particularly used in which type of cases? Where the samples are collected but will not be cultured immediately after collection. So in that type of cases, transport media serve to be very useful. Fine. They provide all the nutrient and environmental factors necessary to preserve the samples and or organisms en route to the laboratory. Okay, clear? It prevents the outgrowth of commensals or contaminants in the collected sample. You should know the names of these type of, you should know some examples. What are they? Ames medium and Carey Blair media. Okay. Then we have storage media. I told you other than studies, there are two other purposes, transportation and storage. We also have storage media. What is that? It includes basic or general purpose media such as nutrient agar in slants or tubes. What is a slant? Tubes you all know, right? Test tubes like this. Slant is what? If you just solidify the agar, keeping it in a slanting position like this. Okay, this is your slant. Slants or tubes are used to preserve and store microbial cultures for long period until they are needed for formal or further laboratory investigations. Okay, chopped, cooked meat broth and egg saline medium for these type of purpose. Okay, these medium are typical examples of storage media used to store bacterial cultures in microbiology laboratory. So this is a compact brief discussion about storage media along with the respective organisms that grow on these media and examples. If you have liked it or have found useful, kindly share it with your friends and stay connected for further class lectures. Thank you so much for your kind attention. See you soon. Goodbye.